Hello everyone, please enjoy this hedgehog before we learn about selective reabsorption. Okay, so you learnt previously about ultrafiltration. So we know that ultrafiltration occurred between the glomerulus and the Bowman's capsule, and it was the squeezing out under high pressure of small soluble substances like sodium ions, potassium ions, chloride ions, water, hormones, vitamins, glucose, amino acids, all those things. They got squeezed out. Okay, so we can see these in our list here. Urea, uric acid, and creatinine are all nitrogen-containing waste products that we produce during metabolism. The only one that you've really heard about before is urea, but we're going to also be talking about how we excrete uric acid and creatinine as we go through this today. Most of the things that we squeeze out into the Bowman's capsule, we don't want to lose. We want to keep them, and so we start trying to pull them back in. This process of drawing them back into our bloodstream is called selective reabsorption. And the majority of it occurs in the PCT, in the proximal convoluted tubule. So by the time we get to the end of the PCT, we've already almost finished almost all of the selective reabsorption. The loop of Henle, the DCT, and the collecting duct are all just fine-tuning. So let's just think about a minute about contextualizing where we're at. So we came out of our Bowman's capsule, and then we went down into the PCT. So it's this bit that we're talking about before we go into the loop of Henle. This is the bit that we're going to be focusing on. So if we were to imagine that we were to cut down here and look down it, we would be looking at something circular. Okay, so this would be the circular outside of our PCT, and we would know we'd have the lumen in the middle, and the cells that line our PCT have infoldings. Excuse this rather terrible diagram. So each one of these is a cell lining the PCT. And then we also know that wrapped around our PCT, we've got loads and loads of blood capillaries. So we could imagine the blood capillaries very close to the edge of our PCT. And this bit here, this is the lumen, this is the PCT, and this is the capillary. So it's really important to have that in your mind to contextualize it. And what we're going to be talking about now, we're going to just imagine that we've flattened this out, but always in our head we know that it's actually a circle. Okay, so we're just going to be looking at an example of one of these PCT cells. So here we go. Take a look here at one of these PCT cells. So we can see, first of all, that these cells have a large surface area. They have extensions on the lumen side of their membrane called microvilli. These extensions of membrane increase the surface area for absorption. We've got all the things that we would expect, the nucleus, mitochondria. Why have we got mitochondria? We've got mitochondria to release energy, and that energy can be used for the active transport of substances back into the bloodstream. What else have we got here? We've also got infoldings on the basal membrane. So we always think about infoldings on the lumen side to increase the surface area for absorption into the PCT cell, but once things have been absorbed into the PCT cell, they also need to get out and get into the blood capillaries, and so these infoldings on the basal membrane also increase the surface area for absorption. Can you also see we've got loads and loads of ribosomes? Loads of ribosomes for lots of protein synthesis, lots of protein synthesis for the production of transport proteins for both facilitated diffusion and for active transport. What else do we have here? Well, you can also see we've got lots of Pinocytic vesicles. We'll talk about this a little bit later on, but basically, sometimes we want to absorb large things. So if we want to reabsorb slightly larger things, we'll absorb them through Pinocytic vesicles. So say we had really small proteins, but they were still too big to come in through a transport protein, then we would use a Pinocytic vesicle to get them in. Okay, so hopefully we've covered here the adaptations for selective reabsorption that we see in these cells. So we would talk about the ribosomes, we talk about the mitochondria, we talk about the microvilli, the infoldings on the basal membrane, It's all classic stuff, really. Okay, I just want to show you this diagram a minute because it shows you a little bit about how the different parts of the nephron look different under the light microscope. So can you see here how the PCT is in orange here, blah, 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 and this little orange square is just showing you that this is a light microscope image of the PCT cell. And if you were asked to identify this as PCT in a exam question, the way that you'd know is you can see it's got these infoldings. Whereas if you were to compare that to the collecting duct, which is in blue, no infoldings. Or the DCT, which is in green, no infoldings. Okay, so the PCT has got lots of infoldings on its surface. And it's also shown quite nicely in this light micrograph image. You can see here quite clearly, this is the Bowman's capsule. And this ball of capillaries, this is the glomerulus. And then because it's a cut through, you're not seeing tubes, you're just seeing circles. So we can see this is the PCT. We can see these infoldings here that tell us it's the PCT, whereas these are smaller and they don't have infoldings. This is the DCT. So it enables us to identify these different parts of the kidney under the microscope. So when we talk about the process of how selective reabsorption in the PCT works, it's incredibly similar to what you already know. So it's basically exactly the same as what you learned about in year 12 when you learned about absorption of digested materials in the small intestine. So not a lot of this is going to be particularly new, but let's just go through this process. So here's the lumen, so where the glomerular filtrate is flowing through. These are the microvilli, the extensions on the cell membrane, and even though they're not here, you'd have extensions on this basolateral membrane as well. So, sodium ions are actively transported out of the basal side of the PCT and into the blood capillaries, and we can see that here. These sodium ions, sodium-potassium pump, we know that 3 sodium ions being pumped out, 2 potassium ions being pumped in. This is active transport, and it requires ATP. This creates a sodium ion concentration in the cell. So there is a low sodium ion concentration in here, and there is a higher, I should have written lower, there is a higher sodium ion concentration in the lumen, and therefore sodium ions are coming in via facilitated diffusion down their concentration gradient. But the sodium ions can only enter via cotransporters. These cotransporters have a secondary binding site for a number of other substances. So here in the diagram, they show the example of chloride ions, glucose, vitamins, but there would also be ones for different monosaccharides and all the different amino acids as well. So let's just remind ourselves about these cotransporters for a second. So here we can see we've got a transport protein embedded in the membrane, and the sodium ion is complementary to one binding site, so comes in here. And the glucose molecule is complementary to the other binding site, so they bind on here. And once they're bound on, this causes a shape change in the transport protein, and the two are pushed out in the opposite side, entering the cell. Now, even though this process is facilitated diffusion, we know that it wouldn't occur without the active transport occurring down on the basal membrane. So selective reabsorption requires active transport. So the sodium ions are entering the cell passively via facilitated diffusion with these cotransporters and drawing in other molecules with them. What effect will this have on the solute concentration of the cells? Well, because we're now drawing in all these solutes, it's going to massively lower the water potential. So this is going to have a much lower water potential in the cell in comparison to the lumen of the PCT. And so water is going to follow passively by osmosis. So water is just going to be drawn in down its water potential gradient, and that water potential gradient is being generated by the introduction of all these solutes into the cell. So once these sodium ions enter the cell, they're being pumped out, maintaining this low sodium ion concentration, and we're getting a build-up of things like glucose and vitamins and amino acids and all these other things in the cell. In the basal membrane, there are specific channel proteins that allow these things to move out into the bloodstream via facilitated diffusion. Once they enter the bloodstream, the blood takes them away, and the fact that the blood is moving away maintains the concentration gradient. So blood which is low in all of these things is arriving, and blood which is high in glucose and vitamins and sodium ions, all those things, is leaving, maintaining the steep concentration gradient. So let's just recap how water is reabsorbed. So as we mentioned earlier, the removal of these substances from the filtrate in the nephron decreases the solute potential or increases the water potential in the lumen. So now there's a higher water potential in the lumen in comparison to the PCT cell, and water moves from the lumen through the PCT cell and into the blood capillary via osmosis. Therefore, as you move along the PCT, the water potential and solute potential, same thing really, the water potential remains the same. The concentration of the solution remains the same. You lose a solute, you also lose some solvent. The only thing that changes is the volume. 85% of water reabsorption happens in the PCT. By the time we get to the end of the PCT, we've also reabsorbed 100% of the glucose. We've reabsorbed 100% of the amino acids, 100% of the vitamins. So we basically do almost all of our reabsorption in the proximal convoluted tubule. The rest is just fine-tuning. Interestingly, what happens with urea is not what you would expect. So we know that one of the main points of the kidney is to get rid of urea, but actually the problem with urea is it is a small lipid-soluble molecule. So it's actually able to move by simple diffusion across the membrane. It doesn't require a transport protein. And because of that, and because it is in a higher concentration in the filtrate than the concentration in the bloodstream, urea is actually reabsorbed passively from the lumen of the PCT back into the bloodstream. Now, this is obviously very annoying, but we don't reabsorb it all. We reabsorb like about half of it, which is why we have to keep sending our blood to the kidneys all the time. Our blood travels through our kidneys like every 8 minutes, and this is because we're constantly trying to get rid of some of the urea. However, some of the other substances are a little bit easier to get rid of. Uric acid cannot travel via simple diffusion, and there are no transport proteins that are complementary to it, so it's not reabsorbed at all. So 100% of the uric acid that is filtered out at the glomerulus is lost in our urine. Creatinine is a slightly more interesting one because not only is it not reabsorbed at all, there are actually transport proteins in the lumen side of the PCT that actively secrete out creatinine into the lumen. So you have active transport of creatinine into the lumen from the PCT cells. So creatinine will be drawn out of the blood capillaries and then into the lumen. So actually, you excrete more than 100% of the creatinine that you filter out in the glomerulus. So say you filtered out 100 molecules of creatinine at the glomerulus, you might add another 10 or 20 molecules in the PCT. So we're actually very good at getting rid of creatinine. Creatinine's a funny one to talk about in exam questions though, and I'm just going to raise this for you in terms of spelling. You might already be noticing that word creatinine, although it's a nitrogenous waste product, it looks really similar to creatine. And you remember learning about creatine and phosphocreatine when you were learning about muscles. Because these words are very similar and they are both specific terminology in your specification, you have to spell them correctly in order to get the marks in an exam. So if you were doing a question on the kidney and you talked about creatinine but you accidentally spelt it creatine, they would not give you the benefit of the doubt. So if you're going to write about it, make sure you spell it correctly. One last thing to mention, although I mentioned it really, really briefly earlier, we talked about the fact that there are small proteins. So not so large that they would have been unable to be filtered out, but too big to be able to be brought into the PCT cells using facilitated diffusion with a transport protein. So these small proteins, how did we say they were reabsorbed? We said they were reabsorbed by pinocytosis. So we know that's an infolding of the cell membrane and then a drawing in into a vesicle. And then that vesicle would move through the cell, and then it would be exocytosed into the tissue fluid, move through the ports in the capillary, and then be taken away. So small proteins are reabsorbed by pinocytosis. Okay, that's it. That's everything that you need to know about selective reabsorption in the PCT. Well done. I'll speak to you soon.